On the morning of June 18th, 2002, a neurosurgeon who had co-edited the only textbook on CCM received an email from a mom in Virginia. She was starting a patient organization and asked for some direction. That afternoon, the neurosurgeon wrote her back and offered to be the new organization's scientific advisor. The rest, as they say, is history. We could not have imagined 17 years ago where we would be today, but I am honored that Dr. Awad continues to serve as the chair of, of our scientific advisory board, and that his clinical and research expertise are focused on our families and our disease. In addition to Dr. Awad's brilliance, his dedication, and the sheer volume of stuff he's able to do in 24 hours, I don't know if you guys know this, but truly, Amy and I just marvel at this all the time. I particularly appreciate his patience with my impatience. There should have been a cure yesterday, we all know this, and his willingness to teach at every level. And by the same token, I appreciate his impatience, which does show itself from time to time. As a physician scientist, Dr. Awad is on the front line seeing patients, hearing their pain, and offering them hope, and he does a very good job of this. It is difficult, emotional work, and I know that he too would have liked a cure yesterday for our families, but we will get there. Although he's still a young man, in the last few years I have started to ask him when he will retire. We all know how difficult this will be to fill his shoes in the CCM world. And as a patient community, we worry. Every time I ask, he tells me, not until we have a treatment. So I am taking him at his word. I do hope that because of that, he gets to retire sooner rather than later because we have a treatment. But I am relieved that he plans to stay for the duration. We started this together, and we'll see it through together. So now I have the honor of introducing Dr. Awad as the keynote speaker for this, our scientific meeting's 15th anniversary. He is the University of Chicago's John Harper Seeley Professor of Surgery. He leads the Neurovascular Surgery program and co-leads the Stroke and Neurovascular Care team. He's the director of our CCM Center of Excellence, as well as the Safadi Center of Excellence in Clinical and Translational Neuroscience, and the Center of Excellence for Hereditary Hemorrhagic Telangiectasia. We're all familiar with his CCM research. Very little happens in the US CCM research world that does not include him. He's held leadership positions in professional societies in the US and around the world, including as president of the Congress of Neurological Surgeons, chairman of the cerebrovascular section of AANS, chairman of the Congress of Neurological Surgeons, and on the executive committee of the American Stroke Association and the board of governors of the American College of Surgeons. Dr. Awad has excellent taste in restaurants and wine, and he has professional level expertise in telling a good story. So without further ado, may I present Dr. Awad.
Good morning. Thank you. I'm truly honored. And when Connie asked me to think about giving this address a few months back, I kind of took a little time to figure out what is the best way of telling the big story, the big picture, but yet position us where we are today on the road ahead. And that's really gonna be the theme of my talk. This paper we wrote just a couple of months ago was meant to be a summary at a level that a basic clinician can understand about a lot of the research that is happening and also the evidence-based. So basically it embodies a concept of taking a disease that neurosurgeons have struggled with on the technical level of treating a patient and removing a lesion from their brain, and really deconstructing it using the tools of modern biology. And this has been really the story of the last 15 years. So what I want to share with you today is basically some glimpses of what my life taking care of CCM looks like, and that's kind of what it looks like. It's got faces of patients, it's got colleagues in the lab, in the clinic, debating what to do about a case, and it's got connection with scientists that we sit down and we huddle together and we ask what can we do together. So I want to start really by reading you a few verses from a poem by Khalil Gibran, written in 1923, talking about what it is to work with love. It is to weave the cloth with threads drawn from your heart, even as if your beloved were to wear that cloth. It is to build a house with affection, even as if your beloved were to dwell in that house. It is to sow seeds with tenderness and reap the harvest with joy, even as if your beloved were to eat the fruit. It is to charge all things you fashion with the breath of your own spirit, and to know that all the blessed dead are standing about and watching. Work is love made visible. So with that in mind, we start talking a little bit about the milestones and the road ahead. It's a story of collaboration, which means working together. Synergy, which is a little bit different than collaboration. Synergy is about working together to create a higher value or a higher product than each alone can do. And then cross-fertilization is even another level beyond synergy, where you create new concepts that would not have arisen alone by any one endeavor. So this is about community. It's about sprouting, just like we talk about sprouting blood vessels. And it is really starting with writing a little brochure in 2002 for patients and ending up with what our website looks like today, with the layers of and dimensions that we are dealing with. As I tell a little bit the story of the science, because this is what the patients want to hear, they want us to explain to them what the science is about, I want to really apologize about really the trap of all keynote addresses. So if I fail to mention all important contributions, please forgive me. It's very difficult to do that in a time like this, and there is absolutely no intent to marginalize or underestimate anyone's work who I don't mention today. So in a brief history of the CCM, this is about a story that has spanned just over 100 years. And in the late 1880s, the pathologists were starting to see this lesion in the brain, and they were giving it different names. And the father of American neurosurgery from the technical standpoint, Walter Dandy of Johns Hopkins, had a drawing in one of his books with his handmade drawing of a histology picture of what the cavernous angioma looked like to him and how it sat inside the brain. And if we go forward till the era of MRI, about 80 years later, we could see that almost that same appearance now with an MRI at high field. And with us starting now to apply the tools of modern biology to dissect that lesion. So really nothing happened until the 1980s when MRI came about and when the lesion could be seen in vivo. Because before that, it was a pathologic curiosity. Once we knew that it was seen in vivo, we knew what the patients were telling us through their bleeds or through their autopsies, but now we were able to see it even as they survived it. We learned that the disease had familial features. We then went on to, with many people in this room, to map the genes in the familial cases, and early natural history studies were written, and I will show you a few of those key papers. Then, in the next 10 years or so, The work got going to try to understand the function of those genes. The concept of permeability disease was presented at one of these meetings here, and many of us came here not knowing that concept and left with an aha moment that this is probably what this disease is about. And again, it was the work of biologists who are in this room. The work on somatic mutations, the immune response, the concept of phenotype rescue, and then the idea of what is a hemorrhage, when everyone is saying, did my lesion bleed, did my lesion did not bleed, and so forth. And then those have led to the current decade where we are really hammering away at mechanisms of lesion genesis. How does the lesion start? How does it progress? What are the biomarkers, the natural history, leading to the threshold of trial readiness? So these are some of the very, very early papers, people describing the MRI appearance, trying to figure out what might this lesion really look like in the brain based on the picture we see on MRI and then what we would see on pathology. The group at the Barrow Institute in Phoenix really led this effort. Later on, other work came up, including work from our group in Cleveland on natural history. These were very early natural history studies where we were trying to tell where are the lesions in the brain? Are they preferentially in one place or another? We put needles in the lesions as the patient was anesthetized and raised pressure and lowered pressure and published these things, and we tried to understand what is this lesion about. Then there was this issue of using the tools of modern biology to try to start to understand the lesion. Put an immunohistochemistry stain on the classical histology and find out that there was angiogenesis, that the cells were actually proliferating in the lesions, and then when we got stains of the junctional proteins, see how those were lost. And then when we looked with the electron microscope and we saw that the lesion actually was a leaky sieve, all of those were very early, rudimentary. I mean, right now we would sit and roll our eyes as to how primitive those techniques were. We even took cells and tried to culture them, but you know, after one particular pass of those cultures, nothing came of it. However, nowadays we are revisiting 20 years later some of these same efforts. Then collaboration started, and we started getting blood from families around the country. And I had the opportunity to work with Rick Lifton, and Murat Günel was at the time a resident with us. And Rick Lifton, who is now the president of Rockefeller University, was the geneticist at Yale. He was head of the program in genetics. And he said, if you've got families, we'll find the genes. And sure enough, we got in the game. And I know there were other people in this room and in France that were all trying to do this work. And within months and weeks of each other, you started to find that there are 3 gene loci that cause the disease, that there was a founder mutation in the Hispanic Americans of Mexican descent. And these were the first fruits of collaboration, because this could not have been done by a neurosurgeon alone or by a geneticist alone. And then something kind of happened at the instigation of Connie, which is, why don't you guys get together and talk to each other? And Doug Marshuk was at the time leading a lot of these efforts, and he called a bunch of people and he said, why don't you come down to Duke University and let's have a first meeting where we all meet and talk about it. And Amy, standing there, was a graduate student at the time, but you know, she was a leader just like she is now, and this is what the group looked like. Many faces are still here who have not missed a single meeting during the past 15 years. the agenda was amazing because in all the work we had done, we've never kind of had a worm guy and the fish guy and the mouse guy talk to a neurosurgeon and a neurologist, and we kind of got ideas from each other, and then we decided at the end of that meeting that we should write a white paper and go to the NIH and tell them that this is a field worth investigating. It took about a year and a half, and then the NINDS workshop on the biology of vascular malformations took place, and the late Bill Young and myself were asked to offer the first talk summarizing AVM and cavernous malformation biology. And out of that meeting, with many of you presented at that meeting, a paper came out that is a white paper about the research priorities on the biology of vascular malformations of the brain. It is sad that Bill is not with us. It was a friendship that was stolen too soon. This is here he is in France, where we went to visit the Sorbonne together, coming to Chicago or biking together in Rio de Janeiro, and then all of a sudden he was not there. However, through his work, the team at UCSF has sprouted with amazing strength and kept the effort going. And the layers of collaboration continued through new initiatives where we are working with Helen and other folks involved in that effort. Collaborations turned into synergy and cross-fertilization. Doug and Mark, I don't know if you remember coming over to Evanston and sitting together because we had the 13 Tesla MRI and we had mice now that developed lesions, and we were wondering what can we do with this information. Most importantly, since Mark Ginsburg knew what those cells did when they connected to each other. So at those levels of think tank communications were happening behind the scenes, and as a result of them, friendships arose. Doug had a room in my house when he came to Chicago, and then Connie and Amy and I got together. We visited each other's labs. I'm trying to illustrate how this type of community, I'm sorry, this keeps popping up, this type of community sprouted. We reached out to the French group, and through a joint grant came the paper on SWI, double the number of lesions than on GRE and T2 star, and also increasing with age. You know, never before had this been done, and this was a collaboration with the Montpellier group and our group in Chicago, and also work in Rio. The Canadian connection from the very beginning was there. The worm was a big factor in it, but the patients were a bigger factor. And it's amazing what came out of that, including papers that continue to come out in high-impact journals as a result of these very first meetings we had together here. Brazil, they do it in a special way always, and you know, amazing, that group motivated again by a patient and her daughter, and by a dedicated doctor, very, very similar to the efforts that seem to happen all over the place. And then this is what it looked like in terms of impact and multiplier effect. We went to the NIH database and we looked at the funding in a number of grants and in US dollars. And you can look at the years, and I know we weren't planning on it, but this is exactly what happened as a result of this cross-fertilization coming together. The number of grants started to come up. They were initially small grants. Then within about 5 years, the grants became bigger grants. and a total of approximately $56 million have already been awarded by the federal government. This is excluding American Heart, excluding Department of Defense grants that are helping fuel the research that is going on. And this wouldn't happen without advocates at the highest level at NINDS, who are our partners. Jim Koenig, who is here, who is our program director, and the NINDS director, who is a neurologist and who understands very well the importance of this. And I keep telling them that this isn't, this money has given them far more return than other 55 million they've spent on other fields. And it is absolutely true, because you look at the amount of publications and key publications and impact on medicine as a whole, and then understanding new medical concepts that arose from cavernous angioma research. So the British group had another very interesting road that they have taken. I joined them from the very beginning. Here I was in London as their guest in 2008, and what came out of the British part is really this other character with a hat over there, and this is Rustam, of course, who gave the keynote address last year. And Rustam is a brilliant neurologist, neuroepidemiologist, and trialist. And when he started thinking with us about cavernous angiomas, I remember sitting and we drew together these circles of what are the different manifestations of the disease. And we wrote this evidence-based paper defining what symptomatic hemorrhage was. Maybe it would mean something. At the very least, it would tell investigators to talk the same language and patients to talk the same language as to when a lesion bled. What we didn't realize is that it would have a very, very profound impact on natural history. So we had long known, this is a graph that shows the rates of bleeds in different studies. And we knew that when a bleed had happened before, the rate of bleeding again is higher. And also in the brainstem, when a bleed happened, a rate would be higher. So there were a lot of people, mostly Canadian colleagues, that say, don't worry, cavernous angioma never does nothing. It doesn't do anything to you. And then other people would say, now wait a second, if you really bled before, it's a very serious disease. And as it turns out, when Rustam looked at the population study, as you know, in Scotland, they have Edinburgh and Glasgow, and they collect data throughout the Scottish population. So whether it be an autopsy or a family doctor or a hospital admission, they keep track of it. And in that particular series, it became absolutely evident that once a lesion bled, it is 10 times more likely to bleed again for the next several years. And therefore, this concept of cavernous angioma with symptomatic hemorrhage is a relevant aspect of the disease that what was previously not known. This led us to sit down together and again at the instigation of Connie and Amy. And you guys disagree too much with each other. Why don't you sit down and tell us what you can agree on? What does the literature really say? So it was about a 2-year exercise of reviewing every publication possibly made in the field and using standards of evidence and also very rigorous methodology to achieve consensus. So we don't sit down and say, you think the way I do and I think the way you do. We actually sent it anonymously till we all had consensus with each other and came up with recommendations. The problem is that even when we agreed on things, they were not based on level A evidence. They were not based on the best evidence that medicine has today, and it left us with the feeling that there is an intense need for us to do better in terms of creating evidence for what we do, whether you should take aspirin or shouldn't, whether you should work out or not, whether you should take vitamin D or not. We just don't have the evidence that is needed to give you the answers with the level of certainty that you deserve. Of course, meanwhile, doing surgery advanced a great deal and helped many, many patients. This is a lesion being taken out from the speech area in a patient with epilepsy. It's not plucking it out. There are arteries that are important to preserve, veins important to preserve, speech function that is mapped. These are complicated surgeries, but they do wonderful things. This person is cured from both the epilepsy and the cavernoma as a result of this surgery. And also, see if we can go to the next slide. Here we go. So the brainstem is a major challenge. We all take a lot of pride when we have these particular perfect results after we go into the depth of a patient's brainstem. However, the details are more complicated than this. And for every one that looks like this, there is another one that is left with double vision, walking with a cane, or having a problem the rest of their life. This is what it looks like when we do surgery as a last resort. This is a lesion that we couldn't reach. It kept bleeding and bleeding, and eventually we had no option. This patient was on a ventilator, was in a coma, and we eventually did surgery. Now she is going to spend the rest of her life disabled, but she will attend her kids' graduations and birthdays, and her life is worth living, but with a huge, huge cost. So the devil is in the details. You know, it's not take it out, don't take it out. And as it turns out, with brainstem surgery in particular, the cost of surgery is very high. These are the series reported from the best center, the best chest-beating Phoenix surgeon, Seattle surgeons, New York surgeons, reporting their best results. 50% of the patients in those series have more serious disability after the operation than they did before. So it is, and you know that in reality in your communities, the results are worse than that. So what have we learned about disease biology, and will this knowledge really help us treat cavernous angioma safer and more effectively in the future? So I'm going to run with you through some of where the science is. It started with the 3 genes, and importantly, one is much more aggressive than the others. One has a founder mutation in Mexicans of American descent, the other in Jewish families, and now we know in mid-American families, and so forth. So this has already, these genetics research things have helped you every day as you sit down with your doctor and make decisions. What do I have? How does it affect my son or daughter? Does it skip generation? Does it affect men and women equally? All of those things we have answers to that didn't exist until we knew that those genes were Mendelian autosomal dominant. Of course, exceptional aggressiveness of CCM3 disease is not only true in patients, it's true in the mouse, it's true in the cell, it's true in the worm, and therefore it may hold the key to trying to understand what disease aggressiveness is mediated by. What is it about this gene that does that? The work of Kevin Whitehead and others showed us that we are lucky that we only lose one copy of the gene. If you're born with both copies gone, you don't even have a heart. Okay, so that you can't live with no copy, with 2 copies gone. So this gene is not a trivial thing that causes an occasional lesion in the brain, go on and live with it, get over it. This gene has a fundamental biologic importance, because if the second copy doesn't happen, you don't even form a heart. So this really woke us all up. There is something more profound biologically here about this trivial lesion than we had ever thought. And then what that meant is, what happens for you to form a lesion? Since every cell in your body has one copy that's gone and one copy that's good, what the hell happens? And then work took place, many groups, dissecting the lesion and finding ultimately that in the lesion both copies of the gene are mutated, so that the lesion looks like you lost both copies, while the body is lucky not to have lost both copies. So these are the papers that started this, and also the work of Amy and Doug showed that it happens with every particular one. Then we started to mine the sporadic lesions that look similar than the familial, but don't have the inherited mutation. And lo and behold, within those same lesions are the same mutations of the familial case. They just happen as a fluke in the lesion. So what that means is that the biology of the gene applies to even the lesions that are solitary. So the familial disease is not different from the sporadic disease, because ultimately the lesion is a genetic lesion, whether you inherit it or whether it develops by a mutation. Those are fundamental findings that led us to develop these animal models. These are real cool models. You know, the mouse brain is the size of a peanut after the mouse has grown up. And with that, we can create cavernous angiomas that look just like yours. And they look small at the beginning and big, and they bleed, etc. And it actually allows us now to be able to look deeper into the lesion and how it develops using those models. And we know that the mature lesion has inflammation, has leakage of hemorrhage, and that's the lesion we see on MRI. But yet there are little tiny lesions you don't even see on MRI that are mere bubbles in the capillaries, and they have no bleeding, they have no inflammation, but they already have the mutation. So these are the seeds of the big lesion. And this is a paleo-CT or a micro-CT. Almost all the papers now, you see these micro-CT pictures. What you guys don't realize is that this paleo-CT is developed to study insect fossils. It is actually in the basement of the Department of Organismal Biology and Anatomy in an ivy-covered building on campus, University of Chicago. I know at Penn it is at the medical school, but our place, they just study insects with it. And we go 2 afternoons a week, my team, with a big plate full of brains coming from Penn, from Duke, and we borrow the scanner from them. And this has revolutionized the field, because we're able to now count how many lesions and compare treatments in mice and see the effects of therapy. So as a result of work like this, that signaling aberration that make you not form a heart, if it happens on a brain blood vessel after you've formed your heart, so you're lucky to be alive, it forms a cavernous malformation. This is the work by Mark Kahn's group. And this master signaling is critical. This means, to my knowledge, this is the first disease ever where the gene is responsible for such a fundamental organ formation, but then after the organ has formed, the same gene in another cell causes a disease. So the downstream signaling of this had been studied by Mark Ginsburg and others, Johnson, Whitehead, and realized that the downstream signaling involves a pathway of activating Rho kinase, which result in unzipping and making the endothelium leaky. So this particular concept led to the idea that if you inhibited the way this comes unzipped, maybe you rescue the disease even if the gene is still bad. So that this is a fundamental concept in genetic medicine. If you cannot change your genes, you can change what they do. So that's kind of what that's about. And sure enough, that proof of concept was true. By inhibiting Rho kinase, the lesions in the mouse do not form as much. These are the early studies based on really just a few handful of mice. And eventually, this led to work where we also looked at how statins, which are the drugs so commonly used, but when you use them at doses with a drug like atorvastatin that can cause Rho inhibition, you can have the same effect as with the ROCK inhibitors, so that when you jack up the dose of statin, it is as if you're giving the drug Fasudil, which cannot be given to a human being on a chronic basis, while statin can. So this has led, after about 8 to 10 years of work, to the actual trial many of you are participating in now, using statin to try to decrease bleeding. Why is the brain a favored milieu? We keep debating this. Although every cell in your body is gone, occasionally we have some lesions in the skin, some lesions maybe in the testicle or liver, but they're not big deal for your life. The big deal is the brain lesion. So why is that? And one of the big issues, a lot of the things we study here, it looks like we took away the cover glass and there are no inflammatory cells. We look at vessels coming and going and forming and unzipping and blood flowing. But this is happening in the middle of a very, very strong inflammatory milieu where there are inflammatory cells that are actually responding to we have shown this, responding to a very specific antigen in the lesion. So it's an autoimmune reaction that is driving this lesion even as the vessel problems are taking place. We have shown that using drugs like the ones you use for psoriasis or chronic ulcerative colitis or multiple sclerosis that are immune inhibiting drugs, using the same drug that is now approved for MS, actually lesions in the mouse do not bleed and do not form as dramatically. So if you look at the fundamentals of what the genes do, you have to look at the thousands of genes affected by the disease gene. So when you have a disease gene, it forms the actual disease by activating many, many other partners. So it's like one bad note screwing up the whole symphony. So then you want to figure out what is screwed up and can we fix other things even if we can't fix the main gene. And doing this took really a huge effort. This is something we spent about 5 years. We collaborated with Brent in Toronto, with UCSD, giving us cells to look at the libraries of these thousands of genes and seeing how are they different in cavernous angioma from normal vessels. And lo and behold, inflammation and the immune response is a huge thing. Even in the worm that doesn't have white blood cells, doesn't even have an immune system, they have genes that are analogs to our immune system, and they're screwed up. Okay? So this is really something that was telling, and what I'm leading at here is that this led us to start to look at these pro-inflammatory states, like when you are vitamin D deficient, you have a pro-inflammatory state, and sure enough, we're seeing now more and more evidence. Yesterday, Kelly Fleming presented data that shows what we showed also a few years ago, that if you are vitamin D deficient, your disease is more aggressive. Now, that doesn't mean I go pop vitamin D, I'm going to be cured, but it may reflect a state of pro-inflammation that had favored disease aggressiveness. Although it doesn't hurt to take vitamin D. I haven't heard anybody getting screwed up by vitamin D, so you may as well bet on it. 24 biologic compounds we looked at, because many of the groups had done research and they said they might be involved in cavernous angioma disease. These have to do with inflammation, angiogenesis, permeability, extracellular matrix remodeling. So we actually did what we call a systematic literature review and said, what proteins in the blood can we measure that people said could reflect cavernous angioma? Okay, we started with that. And when we queried those proteins, looking at their levels, a few of them had some weak correlations. However, when you combine them in a smart way to look at the smartest combination, the one that is the most red, using very advanced statistics that takes every protein, looks at the others, like every protein looks at the others, and say what's the combination that is best, it turns out that a combination of four proteins, two going up and two going down, two inflammatory and two angiogenic, could actually predict whether you're going to bleed next year. That means your body or your lesion are sending signals in your blood today as to whether you are ripe for a bleed. We don't know why. We don't know if you're not eating right. We don't know if your microbiome is screwed up. We don't know if you're smoking dope. We don't know what you're doing. But there is something that is predisposing you to a bleed next year. And the ones that bleed have a different blood work than the ones that don't. This is awaiting multi-site validation, but we've already revalidated it in many different independent samples. Many of you have come to Chicago, given blood, and this is how we've gone up with this. And it's been published in the best journals, Circulation Research, etc. So this is real. Get used to it. This is a blood test. This is what it looks like. Only one of the patients that bled had a blood test that would not have predicted a bleed. Look at this. They separate like unbelievable. Then we also saw that the one has bled a year before also has signals in your blood. So it's not just the one that's going to bleed, but the one that bled already. And the signals from that are a little bit different. Different proteins tell you already bled than the ones that tell you you're going to bleed. So this is the very beginning of smart blood tests, guys. This is not your grandfather's, I'm going to go check my hemoglobin A1C, you know. This is very smart. This is looking at the mechanisms of disease, combining it to try to do an individualized medicine approach. This is emerging from cavernous angioma research, and we're kind of writing it in other medical journals for other fields to apply. We have people in computational biology helping us with how to do this best. And then imaging. And I want to tell you a little bit how we can squeeze information out of imaging. So you look at the MRI, we already know that if you do it really well, like with SWI sequences, you can count more lesions than you can otherwise. So we can tell you more accurately your lesion burden. But has that helped anybody? I mean, you just go out of my office either depressed or feeling better or whatever, but it hasn't really changed your life to know what your lesion burden is. However, if we look at information within the lesion on MRI, one of the ways is to look as to whether the lesion is leaking. How leaky it is. So when we give the dye, the gadolinium, instead of just taking a before and after picture, we take a picture during the gadolinium phase, and we look at the leak of gadolinium with serial pictures, and we do an equation, and we come up with the Scholar map that tells us how leaky your lesion is. And as it turns out, when we look at that, we find that lesions that bleed just increase their leakiness 40%. 40%, while the ones that don't bleed or are recovering, their leakiness remains very steady and, in fact, improves with time. So this is a very interesting measure. It's beyond what you really look at MRI for, and this looks like what the FDA calls a monitoring biomarker, meaning it's a biomarker that can tell you very accurately, very objectively whether you have bled, regardless of whether your doctors disagree or not. Another one we spend a lot of time on, about 10 years worth of work, more than $2 million of research, is on developing the imaging of how much iron there is in the lesion. So if you look at the SWI image, the lesion looks like a black blotch, right? It's like you threw ink on the paper. What we try to do with the QSM imaging is to turn it into a scale that tells us how much ink there is in that black blotch. And that becomes a gray scale instead of a black and white, and then you can turn it into color if you prefer. And as a result of that, we have done very, very careful studies that show that it effectively measures in parts per million the amount of iron in your lesion. So when we take a lesion out from a patient, this is a graph on the left, and we assay the amount of iron in that lesion, it's exactly what the MRI had predicted. So we've shown that in the mice in relation to the blue stain of iron. Therefore, we can measure how much your lesion is bleeding, even when you can't tell on black and white whether it did or not. And that turns out to be another way for us to follow lesions. And as it turns out, twice as many lesions will leak iron than have symptoms during follow-up. So you can have occult bleeds that are not causing you symptoms. We also showed that yesterday in another type of study. That means that we don't have to just do trials on whether you rebleed. We can do trials that tell us whether you're changing the rate of bleeding, and even a 20% change in a rate of bleeding quantitatively can be significant in a trial for us to help choose among drugs, among doses. It may not answer the definitive question, but you don't have to do 1,000 patient trials to answer every single question. And this is what it looks like. This, we published it in the Journal of MRI, again, telling the radiology community that, look, guys, you are sitting on something here that if you measured, look what happens in trial design. You know, instead of having trials that require 300, 190 patients to count bleeds, you could get an effect that is meaningful and significant with 30 patients in each group. And we were able to make this argument to the National Institutes of Health, resulting in $5 million grant to study the statin, using for the first time QSM as a primary endpoint. So this, for the first time, we are going to measure whether you bleed more or less. If it's more, that means statin is bad for you, get out of it. If it bleeds less, that means statin is good for you. So that's what we're going to try to answer. So the list of drugs is amazing. So there are several on the shelf. Some have already started trials. You saw here, statin, Tempol, propranolol. Others are being developed specifically for CCM. When they are in the preclinical stage or first time in man, the road is longer. And therefore, we can try to do more efficient things in between with the drugs on the shelf, but we are awaiting the bigger and better ones, and we are constantly trying to tell pharma that you've got drugs that you've got patents on that can work for CCM. Please spend the money to develop them for CCM. One of those is the multiple sclerosis drug that's already approved by Genentech. They have several years left on the patent, but they're afraid to test it in CCM because they don't want to jeopardize the big market in MS. So believe it or not, that is what we get told. They don't want a negative result in CCM to make them not sell drug for MS. So That's the realities we have to live with, and you wonder why things are not moving faster. The obstacles are different on different parts of this battle. One huge critical gap, I'm getting here close to wrapping up. One huge part of the gap in our advances is trial readiness. Up till 2-3 years ago, different groups would define hemorrhage, would define the endpoint differently. They collect different type of data on patients. And we said, guys, we are one small disease, one small community. We got to all do it the same way. We got together the big players in this group who see a lot of patients, the BVMC group, the Mayo Clinic group, Utah, University of New Mexico because of the Hispanic population, Barrow, and everybody now is engaged in evaluating trial readiness, entering data into the same portal, so that when we are ready to do a big trial, we're going to have multiple institutions able to participate in a way that was not possible in cavernous angioma before. And this, again, belongs to this community. It's several $1 million spent. This is another $5 million grant with the directs and indirects to all these institutions that, as a result, is a gift to the CCM community to be able to do research. The atorvastatin trial is being run on the side of that because we have an opportunity to run it. And this trial involves you, and it is now a third of the way done in the enrollment. Of course, we have follow-up ahead of us. And these trials are amazingly complex. You know, you heard yesterday about the complexity in the Italy trial. Here also it's even worse because we have biomarker and we have double blinding and so forth. So we have safety people, we have monitoring people, the data people can't be us, so we like what data, we don't like what data. So this is a very sophisticated approach, but it's teaching us how to do it. This is something we didn't know in CCM, and now we do it well for CCM. So that's kind of where we are. One of the results of this, again, published recently in Journal of MRI. Do you think CCM would publish in Journal of MRI? So we're publishing because we're able to tell them you can take it to multiple instruments, multiple institutions, and get the exact same result using very specific techniques. So that's important, so that the idea here is that you can deploy these advanced methods at multiple institutions. This is what it's looking like in our enrollment. We're right on the curve. People are, this means webinars, this means wearing t-shirts, this means encouraging each other. Please do not be apathetic about it. Be energetic about it. The answer is worth it, no matter what it is. Yes, good, bad, it's good. One answer that's bad is we can't do trials in CCMs. We're not going to allow that. So everybody should avoid us ending up with an answer that we can't do trials in CCMs. The patients are all coming. They're of all kinds, males, females, ages, etc. We know what they look like. Now, future targets, just a couple of words. The microbiome, who knew that would be important? Until one colony in Mark Kahn's group quit forming CCMs. You know, it was too clean. It was too clean. Then when it got dirty, they formed CCM again. So this led to a Nature paper that shows a relationship between gram-negative bacteria signals from the gut and the brain endothelial cell switches that sense those signals. So it is really a very fundamental story to medicine. Many groups are talking about microbiome, but few have done it with this type of mechanistic impact. Again, thanks to CCM researchers, a whole advance in medicine was opened. Again, where we are now is basically looking at what the normal brain blood vessel should look like on the left. Then we're stuck with the cavernous angioma being a dilated vessel. Then we want to try to understand the switches that make that dilated vessel bleed or not bleed. and in the process even understand switches that could make a normal blood vessel bleed. And we're discovering those as part of studying a cavernous angioma. So a lot of impressive work is going on that the fruits of which are just around the corner. The broader implications are even cooler. You know, I gave a paper yesterday that says some aged brains look exactly like cavernous angioma brain. And I could like show it to a medical student and he wouldn't know if it's a familial case or an aged brain, because cavernous angiomas bleed prematurely, but aged brain bleed. So a lot of the same signals of one apply to all of us as we age. And we talked yesterday about as we get older, all our brains start to look like cavernous angiomas. The big picture is this: teaming up bigger and working together to accomplish greater things. And I want to end with another couple of verses, also from 1923, but this time from the great New England poet Robert Frost. The woods are lovely, dark and deep, but I have promises to keep, and miles to go before I sleep, and miles to go before I sleep. We all have miles to go before we sleep, because brains should not bleed. Thank you very much.